Hello all, Rick Boas here, and today we're going to be discussing the fate of lactate. So where does lactate come from? It comes from glucose. So the reaction here is going to be the same as our last presentation on the breakdown of glucose, except it changes right through here when there is no oxygen available. So as you can see, glucose is going to go through glycolysis, break down into two molecules of pyruvate. This reaction costs 2 ATP, but it generates 4 ATP so that there is a yield of 2 ATP. Now the difference here is when there is a lack of oxygen, pyruvate is not able to convert to acetyl-CoA, and it actually converts into two molecules of lactate. Because there's no acetyl-CoA, the Krebs cycle and the electron transport chain are not activated. So this leads to a buildup of hydrogen atoms. So pyruvate bonds with NADH, as well as lactate dehydrogenase to form lactate. So not only does this get rid of some hydrogen atoms, it also frees up an NAD molecule to accept more hydrogen atoms. So because lactate has these hydrogen atoms, it will lower the pH in the muscle and blood. So eventually it will stop muscles from contracting. So the body goes through the Cori cycle to help flush some of this lactate. So what it does, it takes the lactate, shuttles it across the blood into the liver, where it can go through the second cycle of the Cori cycle. Second phase of the Cori cycle is in the liver, and it's where lactate reconverts all the way back into glucose. So in order for that to happen, lactate first needs to turn back into pyruvate. Again, we're going to need the help of the enzyme lactate dehydrogenase to convert lactate back to pyruvate. Next, gluconeogenesis occurs. This is where the two molecules of pyruvate are able to convert back into one molecule of glucose. Now, this action takes 6 ATP and generates none. But now we have one molecule of glucose that is available for transport. So it re-enters the bloodstream, where it is then transported to active muscles. So next up, we have some questions to answer. So the first one here, how are these pathways affected by exercise intensity and training adaptations? So first off, it's important to note that the Cori cycle is always happening, just like all of our other energy substrates. It just at a lesser degree depending on exercise intensity. So when intensity is high and there's a lack of oxygen, the Cori cycle is very heavily relied on, which means lactate is being shuttled quite often. Now when we're at a steady rate type of exercise, or something that's low aerobic or even moderately aerobic exercise, it's still shuttling lactate, just at a much less degree. I'd also like to note that at rest, the body still shuttles excess lactate to the liver, but instead of being converted into glucose, it actually helps rebuild the liver's glycogen stores. While the Cori cycle is great for the body because it helps regulate the pH and acidity of the muscles and blood, it's still a cycle that we want to rely on as little as possible, because oxidating our substrates is much more energy efficient. So luckily, there are training adaptations to limit this cycle. So there are two main training adaptations in regards to the lactate threshold, both of which are found to occur with an endurance training. So I'd like to note that high intensity interval training is probably a great option as well, since it has been found to have a lot of the same effects as aerobic training. So the first training adaptation is maximum capacity for oxygen. So essentially what that means is with each breath you're able to uptake more oxygen with training. So with being able to take in more oxygen, there's obviously going to be more oxygen available. With this excess oxygen available, we are able to oxidize more substrates, meaning that we can rely on the Krebs cycle and electron transport chain instead of the Cori cycle. So the second training adaptation is an increase in steady rate exercise. So steady rate exercise is essentially you are able to intake as much oxygen as needed for exercise. So once we exceed this level, lactate begins to accumulate. So with endurance training or HIIT training, we are able to increase this level, meaning that we can exercise for longer or at a higher intensity for longer until lactate begins to accumulate. So the next question here is, how do lactate and lactic acid differ? In most research, people use the term synonymously. So when you're talking about lactate, Sometimes you'll say lactic acid, and for the most part that's acceptable. But in reality, these two things are different. Lactic acid is an acid that's created from milk, or lactose, and it also has an extra proton compared to lactate. Lactate is the base that accompanies lactic acid. So the third question is, can lactate be used metabolically? If so, when or under what conditions? So in reality, lactate is a dead end. It itself cannot be metabolized. But as I stated in the diagram, lactate can convert to pyruvate, which is a key ingredient in metabolism. So the cool thing about lactate is it's got several ways that it can metabolize via pyruvate. So first off, like I showed in the diagram, it can go into the liver, where it can be converted back into glucose, so it can be used anaerobically in glycolysis. At rest, obviously, it can help fuel the glycogen stores in the liver. And the real cool thing is that it can, with research showing, it can actually be transported to other muscles to be converted into energy. So not just the liver. And of course, lactate turning into pyruvate, it doesn't necessarily just need to be turned into glucose. If oxygen becomes present again, that pyruvate can immediately go back into acetyl coenzyme A, and then the Krebs cycle and the electron transport chain to produce a lot more ATP. And then we have our last question here. Are there consequences of lactate accumulation on our ability to sustain exercise? What other key factors are at work at the cellular level that could cause exercise cessation? So first off, glycolysis is obviously extremely rapid. It's able to provide us with energy when we're performing quick bursts of energy, like anaerobic exercise. The issue is that it's only producing 2 ATP each time, so we're not getting that much energy out of each reaction. Because of this, obviously our energy demands are going to increase beyond what we're able to produce through glycolysis and the Cori cycle. So because of this, lactate is going to accumulate, which is going to, because of the pH imbalance, it's going to bring our pH down and our acidity up in the muscle and blood. When the acidity rises, the lactate accumulation is going to inhibit some of the enzymes that are needed in metabolism. So this is going to prevent some energy production and effectively stop muscle contraction. So obviously muscle fatigue because of acidity is one of the biggest consequences of lactate accumulation. It's what most people would cite when referring to the research. It's important to note, though, that there are other consequences to lactate accumulation. So if I can bring this up here. So as you can see here in the Cori cycle, we get to ATP, or energy substrate, through glycolysis, but bringing the lactate back around through the shuttle, we have to pay an additional 6, which gives us a yield of -4, or a cost of 4. So because of that, we're going to be losing 4 energy for each time trying to clear the lactate. So not only as when exercise intensity goes up are we going to be needing more energy, but we're actually going to be using more energy trying to convert the lactate. So it's like a doubling effect, meaning that we're probably going to tire much quicker because of lack of energy as well as acid buildup. So that's my presentation, everyone. I hope it was enjoyable and the information was clear enough for you. I'm just going to add here are my references, and thank you so much for watching.